Okay, hi everyone. I just want to go over a couple of topics that we should have been talking about in class. Some of us got to some of these things and some of us didn't. I'll make sure everyone's on the same page. So first off, I want to talk a little bit about energy and energy metabolism as a prelude to what we're doing with enzymes and enzyme function, and especially in relation to the lab that you're doing this week. So first off, there's a lot of terminology here we need to be aware of. Make sure you understand the terms on this slide here. What metabolism is, of course, all the chemical reactions carried out by a cell and/or organism. There's different levels of metabolism, of course. There's cellular metabolism, there's organic metabolism, and they all are interconnected, of course, because an organism's metabolism is dependent upon what its cells are doing and cellular metabolism, right? So they're all linked together. Okay. Most metabolic processes involve pathways. So a metabolic pathway is simply a series of reactions that convert a molecule into another molecule, either converting, for example, glucose into CO2 and water is a series of reactions that would be considered a pathway. And then also taking smaller molecules and building other molecules from that is also metabolic pathway. Synthesis of amino acids, for example, or nucleotides, or all sorts of different things. Right? Those are metabolic pathways, and most metabolism is organized in pathways in cells. There's two main types of pathways you should be aware of. A catabolic pathway is simply the process of breaking down a complex molecule into simpler molecules, and often, not always, but often those are processes that liberate energy. Of course, cellular metabolism, cellular respiration, aerobic respiration, is a process of taking glucose and oxygen and carrying out a series of reactions, catabolic reactions, which break it down into CO2 and water. This is balanced, right? It's not stoichiometrically balanced, but you release energy from that. So that's a catabolic pathway. Anabolic pathways, on the other hand, would be processes that build complex molecules from simpler components, right? And usually those require energy input to do that, because most complex biological molecules are significantly more have energy in them, invested in them, and they're energetically rich. And so building those from smaller molecules tends to require energy input. And a good example of that is, of course, the biosynthesis of protein, for example, from amino acids by dehydration synthesis. That's usually, and that's an anabolic pathway. All right, and then of course, making amino acids themselves is usually anabolic also. All right, so other terms we need to be aware of. Energy is simply capacity to cause change in a system. We think of energy in terms of temperature, work, motion, etc. Right? And different types of energies you should be aware of. Of course, kinetic energy is energy associated with movement. Thermal energy is energy associated with heat, the random movement of molecules and atoms, which is heat. And then, of course, energy can be stored and be in the form of potential energy, which can be released at another time as a result of the system and position of an object. So chemical energy, for example, is a potential energy that molecules hold that can be released as a result of chemical reactions. Okay, so this leads us to thermodynamics, the basic way that energy is, and actually basically the way that the energy is accounted for in the universe, essentially. Two main laws you need to worry about. The first law of thermodynamics is that energy can be transferred or transformed, but cannot be created or destroyed. This means that the universe has a constant amount of energy in it from the get-go. And this is just a basic law of nature that you should be aware of. We're not going to really consider this anymore, except that to know that when we're doing metabolic, to talk about cell metabolism, the first law of thermodynamics is used all the time, that energy is transferred from or transformed from one form to another. And there's always inefficiencies, and some of that energy is not completely transformed or transferred, therefore it is lost. But that energy isn't just disappear, it actually is lost in a form, usually it's heat, it's thermal energy. So that's how you account for that, but it's not all destroyed. So and no reactions are perfect. So there's going to be some usually leftovers, energy leftovers when any type of reaction happens. Second law of thermodynamics is a really important one. Also, every energy transfer or transformation increases the entropy of the universe. Entropy, of course, is the measure of molecular disorder or randomness. You'll be aware of that from chemistry. And the second law of thermodynamics says that the universe is constantly getting more disordered overall throughout time. That doesn't mean there's not areas of local spaces where entropy is decreased, like an organism, a living organism is actually a very localized decrease in entropy because organisms are tightly organized and they're not randomly assortments of atoms and molecules. They're actually tightly organized and they're not very little less entropy than a totally random system. So, but that requires a lot of energy input, of course, and so energy state and flow tends to be related to the entropy of the system in which you're looking at. So living systems tend to have lower entropy, but a lot of energy inputs required to do that and maintain that. If an organism dies, of course, its entropy starts to increase. It starts to decay, to be converted into CO2 and water and other smaller molecules, and the energy is released from that, and the entropy of that organism starts to increase as further away it is from a living state. All right. Okay, now we should be aware of this. The free energy is basically a measure of the amount of energy that's available in a system to do work. So some energy is lost in a system because of the nature of its type of energy it is, that you can't actually do any work with it. But free energy is that amount of energy that actually can be utilized in work in a system. Spontaneous reactions tend to be those reactions that proceed without an input of energy, or they release energy or increase entropy. So one of these two things has to happen. When that happens, you generally have a free energy release, and that reaction is doing work, and spontaneous reactions are of that nature. So if we think about energy in biological systems, we'll have an energy flow idea here, that the first and second laws of thermodynamics come into play when you look at biological systems. Sunlight's coming in from a source, it's an energy source from the sun. The sun is actually taking helium or hydrogen and fusing it into helium. That's its main energy source. That's producing energy that's radiating from the sun in the form of light and a lot of other types of radiation. And that light is coming to the earth, and then organisms on the earth are taking that light, that energy input, and decreasing the entropy of molecules on the earth, which is basically taking that energy and producing molecules in a particular arrangement so that they can be living, so cells can function. That's producers tend to do this. The type of organisms that do this, organisms that do this are photosynthetic. They take light, convert it into biomass, which is living material, usually in the form of sugars to start with, but other types of molecules. And that is that. And when that energy is converted, some of it is not, it's not 100% efficient. So some of it is lost. And because of the first law of thermodynamics, that energy is not just doesn't disappear. It is actually another form of energy. It's lost. That's in the form of heat. So you're constantly losing heat when organisms are doing this. Then you can have other organisms that'll take the molecules that have been produced in this form. This is basically sunlight energy, photons from sun, being converted into biomass. The biomass is now chemical energy, right? Potential energy in the form of chemicals. And that chemical energy can be utilized by other organisms like consumers that will eat that, convert that chemical energy into energy to do work, right? And then the organisms, animals they live, they do their thing. They carry out metabolism, they move around if they move, and they do their thing. And again, it's not perfectly efficient, so you lose some of that as heat. First law of thermodynamics is conserved here, or is followed, and the second law of thermodynamics is followed, especially since this heat that's released from these organisms ends up causing entropy to increase in other surrounding areas, including off the Earth, actually, in the long run. So the overall entropy of the universe is still increasing, even though we have local areas of less entropy here because of energy input, right? Okay, now you should be aware of this equation here. Delta G equals delta H minus T delta S. Delta G is free energy. G is gives free energy. Delta G is a change in free energy in any system. Delta H is enthalpy. They're like kind of whole energy of a system that's in it. And then this is influenced by the amount of entropy change in a system and the temperature of that system, right? So this equation allows us to predict the amount of free energy that's in a system after a reaction has happened or some sort of process has happened. The delta G measures that free energy, and that free energy change allows you to tell whether this reaction can do work or needs energy input to happen, right? Delta G is simply the free energy state of the reaction at the final state versus minus the free energy in the initial state. So delta just refers to a change. Change here is final state minus initial state energy states. So a higher initial state energy state and a lower final state energy state results in a negative term here, negative delta G. And that means that when this reaction runs, the free energy change from initial to final is large or less than zero. That means that delta G here is negative. And that means the reaction will be spontaneous and that you can do some work with that released energy also. All right, here's examples. You can look at these if you want. Someone going off a diving board using gravitational energy, converting that into kinetic energy, and then hitting the water. And of course, their delta G state when they hit the water is lower than it was. Their free energy state is lower when they hit the water than it was when they jumped off the diving board. A lot of their energy is lost. The kinetic energy is lost as heat in the water when they strike it. Also, if you put a pulley or something on this guy's ankle and he dives off the diving board, he could actually run a motor, right? Spin a wheel or something, do some work, lift weight or something like that. So this energy of gravity is pulling, causing a kinetic energy change, and that can actually do work, literally do work, right? Diffusion, you can do work with this also. If you put all the molecules on one side of a space and then allow them to diffuse, this is an increase in entropy now, not so much the change here is an increase in entropy, delta S, not so much a change in enthalpy, delta H, but because this term's negative and this change is basically marginal, this is going to be a negative term. So the delta G value is going to be here is going to be negative, right? So increase in entropy here is going to result in negative delta G value. Again, you can do work, and we're going to see this when we talk about cellular respiration and how much work cells can do with diffusion gradients across a membrane. This is basically proton gradients, which allows cell to make ATP. All right, and then chemical energy, of course, is you take an energy-rich molecule like glucose or something and react it with oxygen. You can break it down into smaller molecules and basically produce CO2 and water or whatever that might be produced, and that will result in a change in entropy, an increase in entropy usually, and also maybe a change in enthalpy also, and release in heat. And so this delta G value tend to be negative also. Okay, so when we're talking about enzymes and metabolic reactions, we need to talk about two main types, right? There's reactions that release free energy as a result of the process. So that reactants start with high energy state and end up with a lower energy state. So when they run, that reaction energy is released, and that would be a negative delta G value, this difference between the two. Remember, free energy state of the final product, sorry, of the initial product would be higher than the final energy state of the final product, right? This is called an exergonic reaction. Exergonic, learn that word, exergonic. This means a reaction that releases energy in the process. This is a spontaneous reaction. It does not require energy input to happen. Doesn't mean it will happen automatically, though. This may require catalysis to make it happen, right, in a normal state. But when it does happen, energy will be released. This usually means the system heats up, but not always. Okay, you have the opposite here. If you have reactants, low energy, and then products with high energy state, to make that reaction happen, you need to put energy in. This is called an endergonic reaction. The delta G change is greater than 0. You need energy input. All right. So classic reaction that we're worried about in this class is glucose plus 6 oxygens goes to 6 CO2 and 6 water. The delta G value of that is minus 686 kcals per mole. This is really, really exergonic. And of course, you can see this in the form of like a marshmallow. If you take a marshmallow, which is basically glucose, it's sucrose, but essentially it's equivalent to being glucose. You can light that on fire. What that does is combines oxygen with glucose and releases heat and light, and produces CO2 and water vapor. And of course, it burns, and that's the energy release from marshmallow. Your cells do this reaction all the time. It's just that they don't burn in the process because they do this in a controlled way. They run this reaction and they take the energy out of this, and they do it in steps so that the energy released in different steps can be captured in some sort of form to allow the cell to do work with it. So we'll talk about that right now. How's that happen? Oh, let's skip this. So this is just more illustrations of what delta G changes mean. This should be pretty straightforward. You look at this PowerPoint later if you want to take a look at this. Okay, so how do cells store energy? This energy is released in these processes. Well, they use this molecule called ATP. Generally, there's actually some other molecules that do energy storage also, but ATP is the big one. We'll hit the other ones later when they're relevant. ATP is a molecule, a monomer actually of nucleic acids, which we have not talked about yet. This is adenosine triphosphate, ATP, right? And so the adenosine refers to the adenine base here, which is one of the bases in nucleic acid, and the ribose sugar here, these two things, adenosine, and then stick on phosphates to that, and that gives you ATP. Now, you don't need to memorize this structure, but be aware of what ATP is basically, and really understand what these phosphates are doing here. These phosphates are stuffed into a small space. They're covalently linked to each other, and a phosphodiester bond here. And these negative charges on these oxygens are really not happy to be right next to each other in this very small space here. So this is a very energetic bond. This thing, if it hydrolyzes, will really encourage that phosphate to leave. So this bond here is relatively easy to hydrolyze. So this is kind of an energy storage. If you get this put onto this, if you get an extra phosphate, the third phosphate linked on here, you're storing energy in that structure. So the cell can move these phosphates around from ATP to other molecules, stick them onto other molecules, and cause things to happen. So the energy that's released as a result of hydrolysis of this bond is actually used to do mechanical work in cells, make proteins do things and so on. Okay, so basically hydrolysis ATP results in release of energy can be used to do work. So here's ATP in a schematic form, and you hydrolyze that to liberate that phosphate, and you get ADP, which is adenosine diphosphate, 2 phosphates, and energy, right? Well, this energy is not this amorphous thing here. It's not just a magical thing that goes around, does things. This phosphate's where that energy is. If the phosphate moves to something else and interact with a protein, let's say, and that phosphate causes the protein to rearrange its shape because it's a negatively charged group, has lots of oxygens here, these are polar, and this thing will really like to hydrogen bond with things, and they can ionic bond with things that have negative charges. So when this is stuck onto another molecule, like a protein, might change its tertiary structure, might change its quaternary structure. This can do work. And if you take that phosphate, stick it onto something, and then cause the molecule change shape, and then take that phosphate and remove it, molecule's back to its original shape, add a phosphate, remove, add a phosphate, remove, add a phosphate, remove. So if you have something doing that, you're essentially causing a change, an oscillation between two shapes. This can function as a pump or a motor or all sorts of things, depending upon the type of protein we're talking about here. So these phosphates are really important for helping molecules do work. All right, so this means that we can also think about some basic thermodynamics here. If we have a reaction that's endergonic, the delta G is positive. So for this example, we're going to make an amino acid glutamine from another amino acid, glutamic acid. And the way we're going to do that is take glutamic acid, add ammonia to it, right? So we're basically taking glutamic acid as a carboxyl group on the R group, and we put in an ammonia group in here, so it has a carbonyl with an amine on it. That makes glutamine, right? All right. So that reaction has a delta G value of plus 3.4 kcals per mole under ideal conditions, right? Very endergonic. However, this reaction is not spontaneous. It's hard to run this reaction in a cell. It's not spontaneous, right? But you can couple it with an ATP hydrolysis reaction. So you can couple it with an exergonic reaction, which delta G is negative. And cells have evolved ways to do this. They have enzymes that will do this reaction and couple it with the hydrolysis of ATP and result in a reaction overall that's exergonic. So ATP hydrolysis forms ADP and phosphate and releases minus 7.3 kcals per mole. So in an enzyme that's carrying out this reaction, it would take the delta G value of this reaction overall plus 3.4 kcal per mole and add the energy released from this reaction, ATP plus water, to make ADP and phosphate hydrolysis reaction. And that delta G is negative 7.3 kcals per mole. And that difference is minus 3.9 kcal per mole, a negative delta G value. So now overall, this reaction is spontaneous when you have both of these happening at the same time in the same enzyme. So overall, this reaction is spontaneous. So cells can actually make this reaction run by coupling it with a exergonic reaction. So you can make an endergonic reaction go by coupling with an exergonic reaction, right? So this is classic. So you're harnessing energy from catabolism for use in the cell. So catabolism, exergonic reactions, energy yielding, the cell harvests that energy, stores it in the form of ATP, then takes that ATP and does endergonic work with it, cellular work, right? And of course makes ADP and phosphate in the process, right? And then you got to reattach ADP and phosphate to each other through exergonic processes, catabolism, to make more ATP. You go around, go round and around like this, and that's what a cell does, right? And it allows the cell to do all its work. Okay, what kind of work does ATP help cells do? You can move things around a cell, phosphate in one state of a motor, and you release the phosphate and the motor will move because the proteins that are involved in the motor change your shape, right? Okay, work, right? You're moving something. How about pumping ions across a membrane from low concentration to high concentration against a concentration gradient? Active transport, right? Put the pump in one state when it has a phosphate on it, remove the phosphate, the pump goes in another state, and it allows the ions to move across the membrane. And of course, I just showed you the example of glutamic acid being converted into glutamine, right? Chemical work, right? Okay. Okay, I'm going to stop there and start another video on energy enzyme functions. So pick that next video up.